All right. Welcome to Delta Leaf Labs, everyone. My name is Elijah, CEO and co-founder. I'm here on a little bit of a quiet evening, and I've got a few minutes, so I want to show you our new Microbial DNA Collection Kits. So the kits will come in a package like this, and we've designed these kits to be able to allow you to easily and quickly collect microbial DNA from a cannabis plant sample, and then send that DNA back to us legally from anywhere in the world. The process only takes about 30 minutes of hands-on time to complete. There is an incubation step that can take between an hour or up to 24 hours, depending on what you're trying to analyze, what particular microbial contaminants you're looking to identify. So let me just go through and show you the components of this kit and how it works and how you can extract DNA from your plants and send them back to us for testing. So inside of the kit, there's going to be a few components. There's a magnetic separation rack, and you're going to have 2 bags of other materials. So we send you everything you need to conduct this, to extract this DNA, except for what you would need for our normal leaf crush kits. So all you need is tweezers, scissors, a paper towel, and some alcohol, or some alcohol wipes to wipe down and clean your tools in between each sample. So it's really simple. The only extra special piece of equipment you need is this magnetic rack and a little tube rack inside here. Oh, actually, we include some gloves for you as well. So you've really got everything you need. So I'll just go ahead and put these off to the side for now. And in here, you will find this other tube rack that has 4 tubes in it. And these 4 tubes are going to be the processing solutions you're going to use to extract your microbial DNA from your cannabis plant sample. In the other bag, you'll have your instructions. Simple to follow, 1 page. It's got a backside, so make sure you read the whole thing before you get started. It's really important to understand what you're doing when you're collecting samples. It's really, really important. There's nothing we can do to analyze a poorly collected sample. We can't magically make DNA appear in there. We can't magically uncontaminate the sample. So it's really important that you read this thoroughly and closely before you get started, so that you understand what you're doing before you start. Cool. I wrote it, so I know what's in there. Perfect. I think we got everything we need. Excellent. What else do we have in here? We've got a collection bag to put your sample inside of. So you can collect a few leaves, you can collect some buds, you can collect some stems, you can collect roots. You can collect DNA from manufactured products such as edibles. So this is a really versatile kit, and you can use this to screen for the presence of contaminating microbial DNA. You've got a little envelope you can use to package your sample and send it back to us. A couple of clean, disposable transfer pipettes. I'm going to go ahead and keep those in the bag. We've actually got a 5th solution here, the wash solution. Our microbial growth buffer, growth media. So this is used for the enrichment step, so that when you're collecting a sample from plant material or from a infused product, for example, that you can grow it inside of this growth media to enrich for the presence of any potential contaminating microbial life. And so that's an essential step to get a really sensitive reading. We've been able to test this on a few different pathogens and determine that this is an extremely sensitive method to collect DNA. So basically, you put your plant sample in here, you mix it with the microbial growth media, seal it up, let it sit for half an hour, 24 hours, depending on what you're trying to identify. So if it's powdery mildew, you only need to let it sit for about half hour. If you're doing things for a microbial panel, that's such as aspergillus, other fungi, then you're going to want to let it sit for 24 hours, just at room temperature, just in your grow room, roughly 23 degrees to 30 degrees Celsius. It's a pretty robust assay in terms of what we can detect and the range we can detect and the sensitivity. And so it's pretty forgiving, but you want to keep it warm. You don't want to leave it in a cold room overnight. So what you're going to do, you guard your plant sample in there, take one of these disposable transfer pipettes, you take out a little bit of your sample, put a few drops into tube number 1, mix the tubes together and go down the line, follow the instructions. There's different steps where you need to mix from one tube to the next, and then at some point you're going to add it to the magnetic rack, which will separate out the magnetic beads we include in the kit that bind the microbial DNA. You use our wash solution, and then you use the final solution in tube number 4 to remove the DNA from the beads using the other pipette. That's for the last step only. You want to get clean DNA that you send back to us. And that's it. Take it off the magnet, add the extra solution, and you've got yourself some purified microbial DNA that you can send back to us. And the process is half an hour tops of hands-on time, whether you're collecting 1 sample or you're collecting 10 samples or more. So it's really efficient and it's really sensitive. And when you get that purified DNA sample, it'll just be like this in a little tube at the very end. Just a tiny purified DNA sample. Put it back into the envelope or put it into the envelope, and then you can put it into a shipping envelope and send it back to our lab.